Hi, everyone. My name is Kylie LaBianca, and I'm a sophomore biochemistry major at UMass Amherst. Today, I'll be talking to you about my research and all about bacteriophages. So what exactly is a bacteriophage? Essentially, it's a virus that infects bacteria. They attach onto a host and inject their DNA into a bacterial cell until it bursts. Bacteriophages are everywhere. In fact, they are considered the most abundant biological agent on Earth. Wherever exists bacteria, there exist bacteriophages. Ever since their discovery in 1917, bacteriophages have been considered as a therapeutic agent against bacterial infection in humans. A decade after their discovery, antibiotics were introduced to the world and took over as the primary treatment of infections. For a long time, bacteriophage research was put to the side, as they did not have much use in the clinical world. However, as bacteria continue to develop resistance to a variety of recent antibiotics, an alternative to antibiotics is gradually becoming necessary, and bacteriophage therapy is among the most viable of alternatives. For this reason, advancements in phage therapy are becoming increasingly relevant in modern medicine to treat infection. My research project involved annotating a specific novel bacteriophage that was discovered on the surface of soil here at UMass. The name of this phage is Microbacterium phage PineapplePizza. Hence the name, this phage infects the host Microbacterium foliorum, a type of bacteria. The analysis and categorization of bacteriophages allows us to further study them for their antimicrobial properties, and new phages are being discovered and annotated every day. In order to annotate this genome, I navigated to the PICAN database, which contains all of the existing information on the individual genes of discovered phages. The summary page for the PineapplePizza genome included its length and number of genes, and clicking on the Genes tab gave its cluster along with data on specific genes starting from gene 1. Clusters are the way phages are organized, and a phage is placed on a cluster based on its similarity to other phages in a cluster. PineapplePizza is classified as a singleton phage, which means it is not similar enough to any other Microbacterium phage to be included in any existing cluster. This made its annotation a little challenging as there were not many other phages available for comparison. The information listed on PICAN was all automatically generated based on data from various bioinformatics software, such as GeneMark, NCBI Blast, and HHPred. My job was to manually verify the start and function of the genes and make changes and notes based on the bioinformatic data available. Shown here is data from Blast and HHPred for gene 12 of PineapplePizza as an example. While I was going along annotating genes, however, I noticed that one phage in particular was popping up a lot in my research. It turned out that a big chunk of the PineapplePizza genome was really similar to that of a phage called Bacillus subtilis phage 529. This phage was among the first bacteriophages ever discovered, and it was first found in soil in 1965. These electron micrographs here of both phages show that they appear similar in structure, with the short tail and arms coming off of the sides. Since PineapplePizza was newly discovered, there is not yet a higher quality projection of it like the 529 one. Both phages are relatively similar in length and gene number, about 16,000 and 19,000 base pairs, and 23 and 27 genes. The picture on the right shows a simple representation of 529 with the genes and numbers that correspond to different structural functions. There is the head at gene 8, tail knob at gene 9, connector at gene 10, lower collar at gene 11, and appendage at gene 12. It turns out that all 5 of these genes share the same function as genes 8 through 12 in PineapplePizza, and the same numbering indicates that they are at similar locations in the respective genomes. The image on the bottom shows a section of a map representation of PineapplePizza with gene numbers and functions. Note that some of the function names in that picture are outdated and we use different names today. Gene 8 is the capsid, which is the same thing as the head. Gene 9 is the tail, which is the same as tail knob. Gene 10 is the portal, which is the new name for connector. And gene 12 is a tail spike, which is the updated name for an appendage. Another interesting thing I found is that when PineapplePizza was discovered here at UMass, it was suspected that both ends of the genome were linked to a terminal protein. A terminal protein helps a bacteriophage start replicating their DNA, and it is found in some, but not all, bacteriophages. This has not been confirmed, but it is confirmed that 529 has both of its ends linked to a terminal protein as well. So the similarity between these two phages definitely suggests that PineapplePizza is bound to a terminal protein. So I just told you all about the similarity between these two phages. The strange thing about this is that these phages infect different hosts, so it isn't expected that those genes would be in the same exact place in the genome. So the big question is, why are these phages so similar? In order to answer this, it might be helpful to consider if PineapplePizza is similar to any other genome, And in order to do this, it might be helpful to get a more detailed electron microscopy projection of PineapplePizza. It also would be useful to experimentally verify that PineapplePizza does in fact have a terminal protein, because that was a big part of the similarity as well. This research was done as part of the phages project. phages is a nationwide undergraduate research project dedicated to contributing to the ever-growing database of bacteriophages through discovery and bioinformatic analysis. It's a great way to obtain real research experience in an academic course, and I am so grateful to have had this experience at UMass. I'd like to thank my professor, Dr. Peter Chen, for being my faculty sponsor, and I would also like to thank Dr. Jessica Rochelot for both of their guidance in conducting this research. Thank you.